Добрый день, уважаемые коллеги. Мы рады представить вам наших американских коллег из Вашингтона, из National Children's Health System, которые готовы поделиться с нами опытом в последних достижениях в области нейроонкологии, в диагностике и в терапии опухолей головного мозга у детей.
Сегодня будет два доклада. Один будет посвящен фундаментальным исследованиям, его прочитает Джавад Назариан, это будет первый спикер. Второй будет более клинический, его прочитает его коллега Юджин. И лектор просил сообщить сразу, что любые вопросы, комментарии, которые будут возникать у аудитории в процессе лекции, могут быть заданы прямо во время лекции. То есть он сказал, что чувствуете себя абсолютно свободно, можете перебивать, он будет только рад ответить на все вопросы. So, Jawad, please, translational findings in brain tumors, please.
Thank you for an introduction. I'm sure they introduced this correctly. My Russian is not that great. But it's a pleasure and an honor to be here. Thank you for coming to this lecture. And thank you, Ludmila and Alex, for your invitation. I would like this to be a more interactive session. If you guys have questions, you may stop me, ask me questions during the talk. Or we will also have a section after the talk, a few minutes for any questions that you may have. I pulled out these slides this morning just looking at the incidence of cancer within Russia and Europe. As you can see, the cancer incidence rate is less than 6 per 100,000 in Russia. This is statistics of 2012. But survival, the mortality rate of this is actually decreases within the Russian Federation, again, statistics of 2012. The question with this slide is looking at cancer incidence within all of the human beings. It includes both pediatrics and adults. I couldn't find any specific statistics on pediatrics, and I think that's what your colleagues here and we are trying to change, that we need to start thinking more pediatric oriented rather than having a combination of both adults and pediatrics. This is a painting. It's about 500 to 600 years ago. Giotto painting is a mother holding a child. It's a famous painting. Most of you may have seen this. What is the problem with this picture? The child is drawn as an adult. This is exactly how the artists thought about children 500 years ago. And so much of this has not changed today. If you think to clinicians and oncologists and researchers, we still think of children as small adults. It's the same biology, but they're just tinier. It took about 1,000 years of painting and drawing when we started drawing children as children, the way that they actually look. But I'm not sure in the eyes of a clinician or a scientist, we do see children as children and different than adults. And this has started changing. It started changing because of some of the advances in molecular biology. This is the mutation burden, somatic mutation burden in adult versus pediatrics. The panel on your left is the mutation burden in majority of pediatric cancers, that you can see that they actually don't have too many mutations. On your right side on the panel, you have the mutation burden of the majority of adult cancers. And as you can see, the number of mutations, somatic mutations, is much higher when compared to pediatrics. And this is because adults have been exposed more to the environmental factors, drinking, smoking, environment, whereas pediatrics haven't. And if we want to develop precision medicine to treating children, we have to focus on the mutation burden that is causing pediatric cancers and pediatric diseases in general. And why is it? What is beyond genome? This is a cartoon. If you cannot read it, it says, if they ask you anything that you don't know, just tell them it's due to epigenetics. And again, it's a new era of the research that we're trying to understand. And the majority of pediatric cancers are actually driven by epigenetic changes. Epi, Greek word, means beyond, above, is above genetics, so the actual genome has not been changed, but the epigenome of the cell has been changed. And epigenomic is important. It plays a very important role in the macroenvironment. The fact that we have cells that can develop into neurons or muscle cells or epithelium is the epigenetic, and again is driven by the macroenvironment that causes differentiation from a single cell, that you can have multiple different cells that are produced, but also is very important in diversification, speciation, and also changes across different species without changing the genome. A very good example to understand the epigenetics is, I'm missing one slide. A very good example of understanding epigenetics is different cell phones that we use. Some of you guys have an Apple, some of you guys have an Android, I don't know what other models you use here, but the hardware is the same, that's the genome. The software is what is different. And many of you walked in today with your smartphones. The smartphone is actually updating the software right now. That is epigenomic, that is constantly changing. And it's the same for pediatric cancers, it's the same for us, that epigenetic constantly changes, and we need to understand that. This is what we studied in the school many years ago, simple form of epigenomics. These mice, they all have the same genome. The mouse on the left has a mutation in AV. This is typically a golden mice, the gold coat color, and has many diseases, including cancer. The mice on the right, again, has exactly the same genome. It has a hypermethylation of the particular gene, the same gene, the AV gene. It typically doesn't have any health diseases. Again, this is a basic of epigenetics that we were taught in the school, but now we know that the concept of epigenomics is more global in the context of the genome. This is the slide I was talking about. Android versus Apple, they're the same genome, is the same hardware, but the epigenomic, the software of the two systems are different. And this is the slide that Lamila loves the most. And what is beyond the epigenome? What actually causes? Why do we need to study the genome, the epigenome, and the effect, the cellular and the mechanism, the organismal outcome of the epigenomics? These are two different species. They have exactly the same genome, but different proteome. And the different proteome that makes them different is exactly because of the epigenome of them. And again, I'm trying to emphasize this because the majority of pediatric cancers are caused by epigenetic changes, not genetic changes. And we need to study this. The question is that, how much do we know about brain tumors? How much do we know about CNS cancers, specifically in the context of pediatrics? Have we looked enough to identify the epigenomic changes? And if we find them in the laboratory, can Ludmila and Alex take them to the clinic, to their patients, and modify those epigenetic changes? And it happens to be actually a very complex area of research. Those of you who want to do research, laboratory research, there's plenty for you to do. Don't think that everything has been discovered. So this is the genome. We think of it of 20 to 25,000 genes. When you apply epigenomic changes above the genome, it gets a lot more complex. But even at the level of the genome itself, when you have alternative splicing and alternative promoter expression, you get about 100,000 transcripts. From 25,000 genes, you actually end up having more than 100,000 RNA transcripts. But at the protein level is where it gets more complicated. You have PTMs, or post-translational modifications, that you can actually have more than 1 million proteins from exactly the same genome. The majority of these post-translational modifications are, for example, phosphorylation that we see that is causing the majority of signaling molecules in cancer, in pediatric cancers, that we have to be able to identify. So to get the laboratory research and the clinic together, there's a series of things that we have to do, which is what we've tried to establish at Children's National, and which is what we are trying to establish with our colleagues here at your university. The basics of doing research, we do mRNA work, whole genome and whole exome sequencing, followed by proteome, which I just mentioned, epigenomic analysis, clinical, demographic, histological, and preclinical in fainted fonts here. These are cell lines, these are mouse models that we develop in order to understand what is actually causing the cancer and develop some therapeutic for it. We should be able to combine all of this. We should be able to combine the genomics, the proteomics, the epigenomics, the clinical demographics are really important. We think that some of pediatric cancers, some of low-grade glioma specifically, they vary from gender to gender. They tend to have a different manifestation in male versus female patients. But again, this is the tip of the iceberg, things that we haven't really fully understood. But combine this with the preclinical models that we have. Again, we have started to develop some of the preclinical models. The fact of the matter is that we haven't done a good job. And I was talking to one of your colleagues this morning about developing preclinical models. Pediatric rare cancers are not treatable because they're rare and because we haven't done a good job for making the models. We haven't done a good job for developing the majority of the data that I mentioned here. And we have not done a good job with combining that. But this status quo should be changed. And your institution is very much in par for changing the status quo that we have now. The idea is that we would combine all the information. We test those in the preclinical test setting. But at the end of the day, it has to be clinically informative. Anything that we do in the laboratory as far as the genomic data, the epigenomic, the protein profiling, has to make sense such that your colleagues in the clinic can take that and make a child's life slightly better. Slowly, we should make pediatric brain tumors a chronic condition such that we can manage them in the clinic. Cure may be a long way away, but they should become chronic first before we can actually optimally cure them. But we need to accelerate discoveries in pediatric brain tumors. And how do we do this? We need to collect samples. I mentioned to you that pediatric brain tumors are rare. They're typically untreatable, specifically brain tumors of the brainstem. And the majority of these children die at home. We have set up a program that we collect autopsies, where we bring the children to the hospital once they die, or to the morgue, we collect the whole brain and we study the tumor. That's the only way to do it. Nothing has changed for the past 45 years. Children with brainstem tumors would receive radiation 45 years ago, and they would die 9 months later. Today, they receive radiation, they die 9 months later. The only way for us to study was to actually collect autopsy, to bring the child to the hospital, to collect the tumor, and to study it. And I will go through this, how we're doing it. And safety of doing biopsies. Again, biopsies for rare tumors that are in a sensitive anatomical location in thalamus in the brainstem has not been historically done. But recently we have started changing our mind about actually taking biopsies of the tumor and studying the tumor biopsy. This is a standard protocol that we have developed for collecting autopsy. And again, we have patients and families to agree to bring the child to the hospital or to the morgue to collect the brain. This is an example of the brain stem that was collected from a child. And you can actually correlate this with the MRI, the exact section where this brain stem was collected. This is an example of the whole brain. 3 years ago, we were collecting only the brain stem from patients who were dying from the IPG. And parents asked us, would you like to take the whole brain? And you would ask any oncologist, they would look at the MRI, they would say the tumor is only in the brainstem, it's not outside the brainstem, it's not in the cortex. So we don't need to take that, that's the normal area of the brain. We said, no, typically we just need the tumor. But parents actually ask us, would you like to take the whole brain and study the whole brain? We said, sure, we take the whole brain and study the whole brain. And this is an example of the whole brain that is dissected, cut through. We freeze some sections and we fix them in the formalin such that we can study with the molecular biology and also histologically with the neuropathologist. This is an example of how samples are processed. Again, we cut this into slices. We have formalin fixed, followed by frozen samples. And then punch cords are taken for a frozen sample, for example. If you take a punch cord from a frozen sample, it's sandwiched between 2 FFPE, formalin fixed, they can be studied by a neuropathologist. Then we can do molecular studies of the frozen sample to see if what type of tumor does it have. So we've developed a biobank, and this is the biobank that we've developed since 2008. We literally started with 12 samples. These are pediatric brain tumors, not just the IPG, all of pediatric brain tumors. And we have over 1,000. Actually, our database now has 1,200 patient samples in the bank. This is over 4,000 tumor specimens that we have in our bank. And our center is so much smaller than yours, and we see so many less patients than you see here. But we've been able to develop a large biobank that we are studying. And some of the studies that have come out of our biobank is, for example, is that DIPGs, diffuse intrinsic pontine gliomas or brainstem gliomas, are hypomethylated. This is again the epigenetic changes that I mentioned earlier across genome at the DNA level. The tumor cells are actually hypomethylated, and a hypomethylated genome is typically a genome that is open for translation and for transcription. It's unregulated transcription and translation. And this is the mechanism of hypomethylation. The mutation is actually in a histone gene. This is a normal mechanism of methylation, trimethylation of the protein. So I always mention this. There are two types of methylation that we talk about here, two types of epigenetic methylation. One is the protein methylation, which is the histone methylation. The other one is the DNA methylation, and both are epigenetic regulators of the genome. In this particular genome here, this is a healthy genome. The histone genes are being methylated. There is a methionine exchange for the lysine residue, the complex that actually does trimethylation that induces the epigenetic changes. It gets stuck on methionine because methionine has a higher affinity for the EZH2 complex. EZH2 complex gets stuck on the methionine and cannot move. So you get a global hypomethylation in the genome of the IPGs. And hypomethylation, of course, will induce unregulated, dysregulated gene expression. And because of these recent studies, the majority of, for those of you who are not familiar with the field, the majority of data that has come out for brainstem gliomas and for DIPGs really came out in the past 4 years. 5 years ago, we didn't know almost nothing about the genomic of the DIPGs. This is very recent data that has come out. And as a result, WHO has actually reclassified this. This is a new class of cancers that are diffused growth pattern. They are midline location, as shown here by the MRI. And they typically have K27M mutation. And this was actually the first time that we started thinking of pediatric brain tumors different than adults. In 2009, when I was doing the IPG research, kids who were diagnosed with the IPG, they were put on the same drug that adults with brain tumor were being put on. In the past 7 years, the two things has changed. One, we know that pediatric brain tumors are very different from adults. The second thing is that there is location distribution of mutation in pediatrics. This is again is focusing on the IPGs, but brainstem is different from cortex, is different from cerebellum. If you're dealing with a particular tumor in a child, you actually have to look at the location of the tumor because the mutation distribution within the brain is different. So again, important thing to note. One is that the first thing is that we know that pediatrics are different than adults. The second is that within the pediatric brain tumors, location of the tumor is different. This is the distribution of the mutations that you see. Midline tumors typically have histone mutations, K27. This is a mutation as put at the position of 27 of histone gene. Whereas you can also have mutations of histone, but at the G34. If you have histone mutation G34, these are typically cortical tumors. They are not brainstem or midline gliomas. You also have mutations that are shared with cortical and brainstem. This includes TP53, ATRX, DAX, FOXG1, and MYC. And these are, again, mutations that can either happen in the cortex or can be shared with the brainstem. How do we study this? Majority of you guys know, your colleagues, typically your neurosurgeon colleagues, will take a sample, is is analyzed by a panel that is looking at mutations for the cancer, or a sequence to look at the mutation. But if it's a brainstem tumor, unless you're taking a biopsy from the tumor, you won't know the exact mutation of that particular tumor. We've shown in our review paper that what are some of the major mutations that exist in our patients, but then the question was that this was actually one of Dr. Huang's patients who Dr. Huang will be giving a lecture after me. This patient had a metastasis. This was a brainstem tumor that metastasized outside of the brainstem to the cortex, to the ventricle. The question was that when the tumor is leaving the brainstem and invading different parts of the brain, is it the same tumor or is it changing? And why is that important to know? It's important because if the tumor is evolving, is changing the mutation profile, they need to have multiple different drug modalities in order to target that tumor. So I mentioned to you that we were collecting the whole brain as suggested by parents. We took the whole brains and we looked at across the brains. This is an example of the brain, is a schematic of different samples that we took. So we took 9 whole brains and we took 20 samples from each brain. That's close to 200 samples that we collected from 9 whole brains. And the idea was that if the tumor is metastasizing to the cortex, is it actually changing its genome or not? And again, this was done by looking at the genome, looking RNA, protein, and histologically by the help of a neuropathologist. This was important because we wanted to know if we take biopsies of the core of the tumor and the tumor has metastasized, as I showed earlier for a case from our colleague, is the tumor the same? Has it changed its mutation profile or not? Again, example of how the punch cords were taken. This is actually the same patient's brain that was donated. We take a punch cord from this. It would be studied histologically. We would study the genome and protein and RNA transcription of the sample. And we would ask, if you go across the brain, different parts of the brain, again, the same, this is an example, this is actually the brain of the same patient that we collected at the autopsy. The idea was that could we go around this, have it studied by neuropathologists and molecularly. This is the data that pathologists gave us. If you look at the pons, hippocampus, ventricle, the 2 ventricles, and the thalamus and frontal lobe, yes, you see some tumor infiltration in some parts, and some parts are perfectly normal according to histology. Again, this is a neuropathologist taking the samples and saying, yeah, these are fairly normal. But we had a sensitive antibody for histone mutation, and the areas that were fairly normal, if you look at them, you actually have infiltration of tumor cells. The tumor cell that does actually infiltrate throughout the rest of the brain. This is at the time of death. If you have a child who is diagnosed with DIPG, if you look at throughout the whole cortex, you can actually detect tumor cells that are infiltrating the cortex. We took the samples with the mutation analysis, histopathology, methylation, and mRNA profiling, and what we found is that about 25% of the patients, they had metastasis to the cortex, 29% have to the cortex, and 50% of them had metastasis to the cerebellum. Again, this is done by genomic analysis and histopathology. If you would just give the same sample to a pathologist, they would score this less than the percentages that we have. In a pathologist's eye, a very small number of these patients would have metastasis extra pontine areas. And we look at the tumor evolution. This is looking at the pontine tumor that has metastasized to the thalamus. This is an evolution tree. What we found is that this tumor in the beginning, when you only look at the pons of the patient, you only have 2 different mutations, histone 3 and ACVR. As the mutation expands and grows into the thalamus and grows down to the medulla, the tumor picks up extra mutations. You actually have MAX mutation happening in the medulla that wasn't previously present in the pons, and PIK3CA present in thalamus, which wasn't previously expressed in the pons. So the question is that is the tumor metastasizing? As it's metastasizing, is it picking up new mutations or not? The answer is yes. But is it important for therapy? Probably not. because the majority of driver mutations, which in this case are histone 3 mutation and ACVR, are present throughout the tumor. The tumor never loses the primary driver mutations. Regardless of where you find the tumor cells, they have histone 3 mutation and they have ACVR mutation. They may pick up 1 or 2 extra mutations, but they're not clinically relevant. And again, this is the PON of the same patient. We asked the question, if you look at single nucleotide polymorphism, does the tumor change? Again, within the tumor, all of the tumor has histone 3 ACVR mutation. Across the pons and the medulla, the tumor profile doesn't really change much. This is important because it means that if you take a biopsy of the tumor and you profile the mutation, you generate the mutation profile of the tumor, that's exactly what is going to be in that tumor regardless of where it is. In the clinic, you can target histone mutation and ACVR mutation, and you could probably effectively treat that tumor. It's not evolving too much to picking up novel mutation that can drive the tumor differently. And we've shown this again across. Some of the evolutionary trees that we developed for this tumor is that they typically pick up histone 3 mutation, they partner with PPM1D, or partner with TP53. And again, another important observation, because histone mutation needs a partner, it always needs a partner mutation that is either P53 or PPM1D, which both are they're involved in the cell replication cycle. We did not find any single tumor cell that has both mutation. It either picks up PPM1D or TP53, which is again, the importance of this is that in the clinic, if you want to target this tumor, you need to target cell cycle and you need to target H3F3A, histone 3 mutation. And again, mutation profile across 2 different tumor samples. I apologize for this not being legible, but we mapped this. The take-home message from here was that the tumor does not really change much. You have major mutations, driver mutations that you can target. We divide this into 2 groups. They're main drivers. These are the ones that should be targeted in the clinic, and accessory mutations. Are they important for tumor growth? Probably, but are they important to drive the tumor genicity? No. If I wanted to target this in clinic, I would probably go after the main driver mutations, which tend to stay in the tumor and do not leave the tumor as it migrates across the whole brain. So now we know that tumors don't change. If you take a biopsy of the patient, it's probably representing what the tumor is saying. But one of the problems that we had with our studies was that we are always looking at post-mortem sample. What if we take an upfront tumor, a tumor of the patient that has never seen radiation or chemotherapy? Would we be able to find the same driver and partner mutations? There was a clinical trial launched by our colleagues at UCSF. The Children's National is also a partner. In our 2 institutions, we're taking biopsies from patients. This is a child diagnosed with the IPG. This is transcerebellum biopsies. A surgeon sends a catheter through the cerebellum, take a sample of the tumor, and we study the tumor, again, as shown here in this radiographic image of the patient. This is the outlook of how the trial is run. I won't bore you with this, but this is the area that I will be focusing on. We take the biopsy samples from the patients. We develop xenografts. These are mouse model, PDX models. We develop tumor cells, and we analyze them by whole genome sequencing. Again, the idea is that if you have, in your patient cohort, you may identify different mutations, but you can only take 4 FDA drugs per patient to try to look at response to treatment. We take the cell lines, we develop them in the mouse or in dish, and we can put the mouse under a variety of different variety of drug treatment to see if we can actually target the tumor. And we have an active area of research in our lab. This is a mouse model of the brainstem glioma. We have a neurosurgeon who works with us. She takes the mice and she puts in a pump within the brainstem. This is a mouse that is bearing the same tumor as a human. And the idea is that if you put in the therapeutics in this pump, which is shown here, this is a subcutaneous pump. which injects the drug into the tumor for 2 months continuously. If you directly inject the drug into the tumor, do you actually see a quick response because you're not going systemically and it's direct injection into the pons. But what are the challenges? The challenges are with DIPG and the majority of brain tumors is assessing response to treatment. We actually have to go into the tumor and take a biopsy. And in the US, there are 2 different institutions, Children's National and UCSF who are doing this. Other institutions are not. When a child comes in and radiographically is diagnosed at DIPG, that's pretty much it. And in many hospitals across the world, it's the same thing. If you don't have the expertise of a neurosurgeon to take a biopsy, the patient is almost always diagnosed radiographically. There's no other way of taking a biopsy. So here comes the era of liquid biopsy. The liquid biopsy takes advantage of the fact that tumor cells, so this is standard biopsy. The liquid biopsy takes advantage of, if you have a tumor cell that is growing really fast in your body, these cells should be shedding and releasing information. Genes, proteins, RNA, kinases, different metabolites into the bloodstream that you can test. You can look at the CSF or you can look at the plasma and detect them. We've always knew that that's a fact, but we just haven't had the platforms to detect them. The platforms have not been sensitive enough to detect. So we've tested this. We've shown before, we've taken CSF before from the patients, and we have shown data that you can actually detect proteins. But to detect proteins, you actually need to take a lot of CSF samples. And many of the DIPG patients, you don't collect CSF from them. Plasma would be much easier, but CSF is almost, in majority of cases, it's not collected because it's not, it could be dangerous for children. So the question that we were asking was that, how is DNA is being released, nucleic acid is being released into the plasma or into circulating bodily fluid, and does it have long enough life that we can detect it in the IPGs? Why would it be important? It would be important for diagnostics, for personalized therapy. Why? Because the IPG patients may have different mutations that we can detect them, and you can put the patients under therapy that is specific to their own mutations. Monitoring response to therapy, again for cancers that recur, that have high chance of recurrence. Instead of doing an MRI, you could take the plasma and test this. And this has been shown in adults before, in adults. This is a published study that shows in adults that this is liquid biopsy. The blue line here shows that the tumor is actually growing, and the red line is a blood work that was being done. Typically, this is what they do in the hospitals now in many countries. Many hospitals, they do a blood test, look at kinases, look at different markers that may correspond to tumor growth. Of course, there's MRI there, but if you look at the blue line here, which is the liquid biopsy, looking at tumor-specific DNA, they saw a growth that was actually correlating to the MRI of the patient. And in this particular patient, the drop in a tumor burden was perfectly correlating with the drop in the CSF, circulating DNA in the CSF. I've spoke to you before about histone 3 mutation and the role of histone mutation in DIPGs. We showed that patients who do have histone 3 mutation, H3 or H3-1, they fared worse, they have the shortest survival life. So our question was that can we actually detect histone 3 mutation in the plasma of patients? This is a platform that we have that your colleagues also have here. It's called digital Droplet Pcr. It's actually a very interesting, very sensitive platform. It develops these macro bubbles. Within each of them, there's a PCR reaction done. It's highly sensitive. If you have 10 copies of a mutant gene, in the presence of 1 million copies of the wild type, you can actually detect them and count them. So you can calculate MAF, mutation allelic frequency. You can just take plasma, do ddPCR, digital droplet PCR, and understand what is the allelic frequency of the tumor, hypothetically. So we wanted to do this for the IPG. And again, the important thing of this platform is that it's so sensitive that you can target this for a single nucleotide change. Remember, the tumor genome for DIPG specifically is exactly the same as the wild type. There's only a single nucleotide change that we should be able to detect. So we set to test this, and we had the list of genes that we wanted to identify. Again, these are genes that we were interested in that were either involved in low-grade gliomas or DIPGs. And we test them. This is the output of ddPCR, that you can see the wild-type copies and the mutant copies. And you can calculate this. These are the empty bubbles that were generated by the machine. So you can actually take plasma from the patient or CSF, subject them to the ddPCR, digital droplet PCR, and count how many times do you actually see the mutant copies. We've shown that we can do this for H3.3, for H3.1, ACVR, different forms of ACVR, and PPM1D. And again, the detection is so strong that you can perfectly calculate the mutation allelic frequency just by taking a plasma. This is the plasma that you need for this is about 2.5 ml of whole blood, and the plasma that is resulted from 2.5 ml of whole blood. And the experiment actually takes about 4 hours. Within 4 hours, you can detect the mutations that the patient has. And it's relatively cheap. It's about $10 to $20 per sample for such analysis. But also what's important is that, you remember I talked to you about the driver mutations and partner mutations? The question was that, can we do multiplexing? Because again, you're dealing with patients, pediatric population, you cannot keep taking plasma or blood from them. Can you actually take one plasma and look at 2 different mutations, multiplexing of them? And you can actually. In this particular patient, you see ACVR1. This cluster is ACVR1 mutation and histone mutation in the presence of both ACVR and histone wild type. You can count the bubbles that have H3 mutation. You can count the bubbles that have H3 wild type and calculate the mutation allelic frequency, which is again very much correlative to the allelic frequency of the tumor itself. CSF is such an amazing repertoire for assessing assessing ctDNA, circulating DNA. It is compacted and full of ctDNA that you can easily assess and analyze. An example of mutation screening that we did was for this patient, 16-year-old, and had clinical symptoms of lethargy, vomiting, headache, astrocytoma grade 3 was thought. And this is the MRI of the patient. We took the CSF of the patient, and we showed that the patient does indeed have histone 3 mutation. And again, this patient was not subjected to the direct biopsy, obtaining biopsy. And the platform is so sensitive that we are doing this for many platforms. We get samples from Israel, from all over the world, that send us for centers who cannot actually obtain a biopsy from the tumor. And within 4 hours, we can tell them what type of mutation the patient has. And again, you can compare this. You can detect this either in plasma or CSF. Again, CSF is such a much better environment to study the mutation, but you can also detect it in the plasma. These are the cohort of the patients that I mentioned. We have looked at the CSF, and we look at the plasma, the amount of the amount of the ctDNA of the histone mutation that you can detect. And again, in almost all of our patients, we can detect it either in CSF or plasma. But is it relevant is it clinically relevant? Does it actually make sense? If you calculate the multiple mutation allelic frequency of of histone, does it correlate to the patient outcome? It actually does. These are the cohort of patients who were subjected in the PNOC trial for actual biopsy. We took plasma of these patients. This is at the time of biopsy. You can detect a large number of histone mutation in the plasma. It drops post-radiation, which is expected because almost 100% of the children respond to radiation. The tumor shrinks. So the number of histone mutation drops in the plasma and stays relatively low post-radiation, and this was at the end of the therapy. It stays low. If you look at particular patients, this is an example of a patient. This is the amount of ctDNA in the plasma. Patient received radiation and it dropped. It stayed low during the first phase of treatment when the patient was receiving panobinostat, peaked as soon as the study was ended, and dropped again when the patient was put into the second trial. It actually correlates with the clinical outcome of the patient. But more importantly, we took this data and we asked a radiologist if they could draw the tumor size on the course of the treatment. The red lines here were drawn by a radiologist, independent of our study. The radiologist did not know about our study. She scored this tumor size, which is shown here in red, and in black here you have the amount of the DPCR, the mutant DNA that we detected in the blood. It perfectly correlates. Again, the patient received radiation at this point. It perfectly correlates here. We had one patient that the DPCR did not correlate to the MRI. The MRI was showing a growth in size, while the DPCR was showing a decrease in size. This was actually a pseudo-progression. This patient was responding to treatment, but had a pseudo-progression. The MRI showed the tumor growth when it actually wasn't tumor growth. So the plasma liquid biopsy is so much stronger than actual MRI. I mentioned earlier that we need to integrate the data if we want to cure these patients quicker and faster, and we can't do it by ourselves. It has to be a collaborative effort. We've established a biorepository. We have established a transdisciplinary team and cross-institutional team, but because pediatric tumors are rare and because they have so few mutations, it takes, and because we just don't have too many of them to do large trials, we need to be doing this together across the globe. This is some of my team members who work on the data that I showed you today. Some of them are neurosurgeons, oncologists, graduate students, and technicians, a clinical coordinator who has done a great job, and some of the sites in the world that we're collaborating with. Again, the mutation is the same in patients regardless of which country they're from. And the idea of us being here is hopefully to set up a collaboration, to have a star and your wonderful country on the map to form this collaboration. I think there's so much room. Your team is very enthusiastic. We look forward to it. And a lot of thank goes to the wonderful, enthusiastic, and awesome team who are here. And I really thank you. I think this says thank you very much. I had to Google translate it. I'm not sure what it says. Actually, I cross-checked it with Spanish and Italian to make sure it says thank you very much. And our team and the group who's been funding us, the majority of these are families who are mothers and fathers who've lost a child to cancer. So thank you for your attention.